So for part one of this Enzyme Kinetics lab, we're going to be working with an enzyme in a 1 mg per mil solution. This is alkaline phosphatase. And the other thing that we're going to be working with is the substrate for the enzyme. This is nitrophenol phosphate. This is in this 50 millimolar amidol buffer. And the other things that we're going to need for this lab is a multi-channel pipette. We're going to be using a 96 well plate, a 96 well plate reader, and then we're going to have our solutions that we've actually diluted to work with. So these are stock solutions. For our substrate, we have a 1x buffer solution that we've made. We've diluted this 10x stock to our 1x working solution. And then we have done a serial dilution of our enzyme where we now have 250 microliters per mil, 100, 50, 25, and 10. And so what we're going to do is we're going to mix our enzyme and our substrate in different concentrations of the enzyme and figure out the optimal enzyme concentration to use in the next part of the lab. Okay, so we've loaded our substrate and our enzyme into our 96 well plate. And so if you look right here, we have lanes A, B, and C, and we filled them 1 through 6. And so in wells A, we have 250 microgram per microliter solution, and our 100, 25, 50, 10. So we're doing our dilution of our enzyme this way. A6 has 0 enzyme, it's just water, so that's our negative control. B1 through 6 and C1 through 6 each have 240 microliters of our substrate solution. And so we're going to run this experiment in duplicate, so we're going to do 2 tests. So we'll pull this enzyme from well A into well B and read it, and then we'll pull more from well A into well C and read it that way. And so when we do this, we're going to make sure we leave it separated until right before we're going to do our measurement, because as soon as we put our enzyme in, it's going to start working. We want to record that. So we're not going to mix these at all until right before we're going to load them in our plate reader.
So before we take the absorbance of all of those solutions inside the 69 well plate, we have to mix the enzyme into the substrate prior to the reading. So here I'm going to extract 10 microliter of the enzyme in the row A into the row B and the row C sequentially. So here I'm gonna use the multi-channel pipette, setting to 10 microliter. Extract 10 microliter of the enzyme from the row A into the row B. And gently mix for several times. So after that, we put the plates into the holder right here. And this is a machine for us to do the recording. And I click the OK. Now the measurement for the absorbance is starting. It will take about like 3 minutes.
Okay, so the run is over now, and we can see that we have data for our wells in B. And again, our dilution is from 250 microliters, 125 going down this way. So we can see our negative controls all the way down here, and our highest concentration, we have data up here. So that's our first run. So now we're going to repeat that. We're going to mix the enzyme with our substrate in well C, and we'll get another duplicate sample from that.
So now I'm going to add the enzyme from row A to the row C right now. Same procedure as previous. So 10 microliters of the enzyme solution from row A. And then I will insert them into the row C. Gently mix it. So after that, it's ready to take the measurements. So now the measurement of the wafers is starting.
Okay, so in part 2 of this lab, we're going to do a very similar thing that we did in part 1, except we're going to see how inhibitors affect enzyme function as well. So we have a lot of our same stocks. We have our enzyme here, we have our substrate here. We are going to use 2 new things, our inhibitors. We have 2 inhibitors in this lab. They're labeled N and they're labeled P. And so we have a 96 well plate laid out in a very similar way that we did it before. And we followed this template that's in the handout as well. So for our lane A, we're going to look at the spontaneous hydrolysis of the substrate. So how much does it get hydrolyzed even with no enzyme at all? In lane B, we're looking at how well the enzyme works with different concentrations of substrate. And then in lane C, we're going to look at how the inhibitors affect the enzyme function. And so we have everything in lanes A, B, and C, and then we have our enzyme down here in lane E. And we'll follow a very similar protocol as before, where we'll take our enzyme and add it into our solution, and we'll measure it again just like we did before.
Yeah, so soon as the week 1, prior to the measurement of the absorbance, I need to add the enzyme into each row. So now I'm going to take 6 tips at one time. So we have already put enzyme into the row E. Ten microliters extracted, and I put them into row A first. Gently mix. So now from the rest of the enzyme of the lane E, microliters of them are extracted. I pass them into the row A. Gently mix them up. And now it's ready to take the absorbers.
Okay, so after this one is done, we're going to do the exact same protocol that Lydia just showed you with lanes B and C, and we'll be able to look at how the substrate is hydrolyzed without the enzyme, with enzyme, and then with inhibitor. And so we'll repeat this same protocol 2 more times, and all the data from this run will be provided to you.